It takes a village to construct an atlas. HubMap is collaborating closely with many consortia to construct so-called ASCT+B tables that characterize major anatomical structures. They also describe what cell types are commonly found in these anatomical structures and what biomarkers are used to characterize those cell types. So you have experts across consortia, across different organs, agree on names for AS, anatomical structures, CT, cell types, and Bs, biomarkers, which could be genes, proteins, lipids, metabolites. And then having those tables, we are also interested to describe the 3D space that these elements occupy. And so we have constructed, in collaboration with illustrators and also with NIH experts, a so-called 3D reference library, where we now have more than 50 organs represented in 3D. And for those organs, we have major anatomical structures that then serve as landmarks for the registration of tissue blocks. Here you have the spleen of the visible human project male. And if you now have a tissue block that you would like to register because you just took it from another spleen, you would move it to the position where it is inside of the spleen. In this moment, you collide with different anatomical structures. These anatomical structures are then associated to the tissue block, meaning you have annotations for this tissue block. And then via the ASCT+B tables, you also know what cell type populations are commonly found in there and how many of each type. And you also know what biomarkers are commonly used to characterize those cell types. So as soon as you register a block, you not only have annotations for this digital data block, but you also have guidance of what kind of biomarkers you might like to use. For instance, what validated antibodies you can use, or what kind of asymers or other reference you can use to annotate, for instance, single-cell RNA-seq data. So the next step is to connect this evolving human reference atlas, the ASCT+B tables and associated reference organs, to the rich data that HubMap has produced, but also data from other portals, but also to recent publications on atlases for different organs. Once we have constructed the atlas, we can understand how cell type populations change as we age. We can see how they change as we go from male to female or across ethnicities, but we can also see how they change when disease strikes, because we see the differences in cell type populations per anatomical structure, per location inside of the human body for these different conditions.